I'll give it to the security if someone asks. Okay? Yeah. Any questions? Not yet. Not in lecture. Terminal. Terminal? Yeah. Yeah. Position of endospore. Position of endospore. Yep. Go ahead.
Is a negative stain, if it's used as the first stain, is that a primary stain?
We don't use the term primary or secondary because there is only one stain.
Okay.
But
how about doing a capsule stain with the negative stain as the primary stain, as a differential stain?
very good. Yeah, now we are talking. Yeah, sure. Yeah. If you do capsule stain, that's different. Not negative stain. Because negative stain, capsule stain is differential. Capsule stain is a differential stain in which you use cationic and anionic. Yeah.
Okay. In for endospore staining, if we use safranin as a primary stain, then the green pigment.
Yes, smart move. Yeah. I'll explain that next time. Okay, thank you. Yeah. Good question, though. Yeah. Go ahead. Yeah. One more time, sorry. Yeah. As long as, okay, you know what your target is, and if it is darker color, you can use it as a primary stain in a differential stain or a simple stain. What are you asking? Simple stain or a differential stain, right? Different. You have to use the term primary and secondary in a differential stain. So you cannot say any stain. You must use a darker, any dark color stain primarily if it has the affinity for that part. Like malachite green you cannot use for capsule, right? It's dark color. Exactly. What if it is dark color but does not have the affinity for that part? You cannot randomly just say arbitrarily. You can use any dark color stain for any stain. No. Okay? Yeah. Yeah. Oh, yeah. Let's move the test to Thursday. Okay? Yeah. Because I don't think I can. Maybe I can finish it. Maybe we'll stick it to Tuesday then.
No.
No? Okay, fine.
We will all boycott this class and not show up.
All right. That's just fine. I'll still get paid. All right. All right. All right. So exam 1. Exam 1. Thursday. What's the date? 7th. All right. All right. Joshua, do you work on Saturdays or no? Is anybody here on Saturday? Yeah, I'm having a review on Saturday from 12 to 2. Yeah, if you can, no problem. Yeah, so because many of them will not be able to make it to the review, so if you can post it, okay, right here, 12 to 2. I appreciate it. Thanks. Uh-huh.
Should I say that louder?
That's already on there.
Where?
It's already on YouTube.
You didn't,
We didn't send it, but it's on YouTube.
what are you looking up to find it?
Well, if you look up one of his links, all the rest it'll be like on the side. It'll say it'll be like recommended, and that's what it's about.
Oh, thanks, Crystal. I didn't know that. Really? Oh, okay. All right. So, okay. All right. Yes. Yes, I am. Yeah, and we may have it for more than 1 hour because I don't have a class to teach today. So instead of 2:30, we may stay there until 4:30. So it may be 2-hour review today. Yay!
If we give them a little more money, can you get the videos going?
All right. Any questions before we start Chapter 4, finish up the new topic? Any questions? Where did we stop? Cell wall. Cell wall? Okay. We did spheroplast, protoplast, or no? No. Okay. That's where we stopped actually. Okay. We did hypotonic, isotonic solutions, right? Okay. Good. All right. All right. Let me just ask you one question, then we'll move on to the new material. All right. Okay. Let me see if I can ask. Okay. Should I ask for specific people or just general? Okay, fine. All right. So raise your hand if you know the answer. What do most bacteria in hypotonic solution? Proper term. They will become? First answer is rupture.
Plasmoptized.
Second answer is plasmoptized. David, plasmoptized. That's the correct answer. Most bacteria, because they have cell wall, they will be plasmoptized. Okay? They will not burst. They will not burst. In hypo, you see this is your bacteria. After gaining water, it has swollen up. Remember hypo equals hippo? Yeah. Now hippo. Okay. So they will swell up, but they will not rupture. This is how it's going to look like. This is plasmoptysis. And cell without cell wall? Ghost cell. Ghost cell. Good. Name one bacterium, please. Name one bacterium or one cell that will crenate in hypotonic solution. That will crenate. Okay, I hear one answer, mycoplasma. Mycoplasma. B. Plant cell. Okay, Jackie says plant cell. Any other answer? Cheekbone? Cheek cell. You mean animal cells? You mean animal cells. Animal cells. Okay.
All of our, that's human cells, right?
Yep.
All of our cells are...
Animal cells. Okay.
What was your question again?
Yeah, can you repeat?
Which of the, okay, name one cell or cell type that will crenate in hypotonic solution. I have mycoplasma, I have plant cell, animal cell. Any other answer? Not sure? Not sure? Anybody else? Correct answer is D. Bacteria or cells, they gain water but not lose water. My question was crenate.
In hypertonic solution.
Okay? The reason I'm practicing with you is these are on your test. Okay? Yeah, so please, cells do not crenate in, or they don't lose water, they gain water. Okay? So now I can ask you this now. This one you should be able to answer. Hypertonic solution. Hypertonic solution. Which one, name a cell that will become ghost cell?
None.
See, none? Cells don't become ghost cells, they crenate. Crenate here, opposite. In hyper, they crenate. And in hypo, they become ghost cell. Okay? Cells without cell walls. Okay? All right. We'll practice more of this. Let's finish up chapter 4. All right. Any other question while I'm setting up my presentation? Any other questions?
So just to be clear, on the plasma,
Moptysis? Moptysis, yeah. That,
cell with the cell wall will stand up or not?
very good. Yep. Good. Yeah. Plasmoptosis and plasmolysis are the term where both cells, they maintain their shape. Plasmolysis and plasmoptosis is a term for those cells that have cell wall, but they maintain their shape. Plasmolysis, loss of water. Plasmoptosis, gain of water. But in both conditions, cells maintain shape because of cell wall. Okay. All righty. Okay. Last topic about cell wall are the two terminologies, protoplast and spheroplast. Okay, before we left, I think I might have mentioned that these two structures are not found in nature. They are man-made. Oh, nope, not that. Yep, there we go. Okay. Okay. Protoplast and spheroplast, what are they? Okay. They are made in lab for research purposes. Why? Okay. How? First, you need an environment which is isotonic. So, isotonic solution in both tubes. One of the tubes we are going to place a gram-positive bacterium. The other one we are going to place a gram-negative bacterium. Okay. This blue color is going to represent the cell membrane. Both of them, they have cell membrane. Okay. And gram-positive cell wall is almost entirely made up of what? Peptidoglycan. So this red color represents peptidoglycan. Gram-positive. A whole lot of PG, peptidoglycan. Gram-negative, they have only a little bit of peptidoglycan, just one thin layer maybe. Most of their cell wall is made up of what? Lipid, which is called outer membrane. So this green color is going to represent the outer membrane. Outer membrane. Okay. Now we are going to put in each tube a chemical which is surgically going to remove peptidoglycan only. Nothing else but the peptidoglycan. There's a cheaper way. You can simply spit in each tube. Really. Yep. What do we have in our saliva? The enzyme, amylase, lipase. The enzyme we are looking for is lysozyme. Lysozyme. So, or you can add the antibiotic if you have antibiotic that destroys penicillin is penicillin. That destroys penicillin. That destroys peptidoglycan is penicillin. So you can add either penicillin or the enzyme lysozyme. Lysozyme. Both of these chemicals, they digest peptidoglycan only. Okay. So you may need to draw a new diagram in which there's no red color. Red color is gone. Only red color. It's not going to affect the outer membrane. This gram-positive bacterium that has completely lost its cell wall because of chemical treatment is called protoplast. This is protoplast is a gram-positive bacterium. A gram-positive bacterium that has completely lost its cell wall. Protoplast. Gram-negative bacterium which has partially lost its cell wall. And that partial part is what? PG. Only the PG is gone, right? red color is gone. This is called spheroplast. PG. Gone. Okay. This is called spheroplast. 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 Spheroplast, a gram-negative bacterium that has partially lost its cell wall. Gram-negative bacterium that has partially lost its cell wall. In you can say peptidoglycan only, not the outer membrane. That is a spheroplast. Peptidoglycan only, but not the outer membrane. This is a spheroplast. Sure. Spheroplast, gram-negative bacterium that has partially lost its cell wall. Cell wall. In parentheses, peptidoglycan. Yep, go ahead. Okay. Now, we don't use this in our labs, but the scientists want to see the effect of antibiotic on cell membrane. Only on outer membrane. Okay. Or see the effect of osmotic pressure without, how much pressure can these layers tolerate without cell wall or peptidoglycan. All right? So purely for research purposes. Okay?
Is the outer membrane part, so it has the outer membrane,
Correct.
but not,
The peptidoglycan. Which of these two cells, gram-positive or negative, able to maintain its shape as long as the solution is isotonic?
negative.
Okay. Anybody for gram-positive? Pardon me? Isotonic. The solution is still isotonic. Okay, my question is, which of the two bacteria will be able to maintain its shape? Remember, it was rod shape to start with. Which one will still be rod shape after the peptidoglycan is gone? Okay, gram-negative. Everybody, gram-negative. Okay, so neither. Correct answer is neither. Both of them, they have lost peptidoglycan. And last time, the very first thing that I emphasized when we were talking about cell wall, that rigidity of the cell wall is because of peptidoglycan, this thing right here. Yep.
Okay, there's different words going around. I heard this protoplast completely lost cell membrane is what I thought.
I did not say that. Cell wall. Cell wall. Peptidoglycan. Yeah. Okay. Okay. So which of these two cells, okay, will become cremated? Which of these two structures will crenate if I reduce the solute concentration? If I reduce the solute concentration of the environment, which of these two structures will crenate? Both? Neither is the answer. Again, I just asked you the same question I asked there. If I'm going to reduce the solute concentration, the environment will become hypotonic, and cells don't crenate in hypotonic solution. Yeah. 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 Yes. Yeah. 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 Yes. Yeah. I did. See, it would be something if I just asked you this question on the test without practicing. So that's why we are practicing here. No. Even if you miss now, can I take double the points off? Same questions on the test. Okay. So, all right. All right. Yeah. All right. So done with cell wall. Okay. Cell membrane, the innermost layer that all living cells have. All living cells have this layer called cell membrane. Okay. Now, you did not see the first word associated with the first two layers of the envelope. You did not see this word regulate with capsule. You did not see this word with associated with cell wall. This is the only layer which actually regulates. This is the boss that decides what is going to come in, what is going to leave. Okay? It regulates the nutrients and enzyme and other substances that come across the cell. Now, because cells, prokaryotic cells, they do not have mitochondria. No mitochondria. So their ATP synthesis takes place at the membrane level. In prokaryotes, because they don't have mitochondria, and function of the mitochondria is to make ATP, right? Prokaryotes and bacteria do not have mitochondria. So where do they make their ATP? At the cell membrane, site for ATP synthesis. Like glycolysis, Krebs cycle, electron transport chain, they take place at the cell membrane. Okay. Now, all the enzymes that are involved in this process, glycolysis, Krebs cycle, electron transport chain, dozens of enzymes, they're all stored at the membrane level. Last thing, and make sure you understand, you know the term, but there's a question, interesting question on the test. Those cells that are autotrophic, everyone knows autotrophic? Can someone quickly summarize that for me, please? Autotroph. Organic compound. A cell, a microorganism that can transform, that can convert inorganic compounds into organic compounds. Or you can say self-feeder. It feeds itself. It can make its own food. Okay? That is autotrophs. Now, what enables the plants to make their own food, and why can't we make our own food? You sit down in front of the TV with the remote for 10 days. Why must you stand up and eat food? Why? Photosynthesis is the answer. Because plants and autotrophs, they have machinery that enables them to make their own food. By the way, technology is so advanced, I will not be surprised if we have green human beings walking around. Really. You take this chloroplast, you can actually pick your own color. Chloroplast doesn't have to be green. You could be purple, you could be yellow, you could be orange. Have you seen the colorful leaves of the plants? Okay, that's all chlorophyll, different type. So in the future, designer people, okay, here we go. So these autotrophic microorganisms, they have this structure right here. Let's see if I can focus that. All right. I'm going to draw my own diagram. This could be purple, this could be green, this could be orange. These are called chromatophores, little pouches of membranes that are close to the cell wall right here. And these pouches that are called chromatophores, they contain chlorophyll. Chlorophyll. And because of this chromatophore, these bacteria are autotrophs. Okay. Let me draw my own diagram. Okay. Brand new. Okay, fine. Cell wall. Cell membrane. These are, each little pouch is called chromatophore. Chromatophore. Or you can actually call this the dish for the bacteria that receives the signal, just like the dish that receives signal for your TV at home. This is the dish that has what in it? Chlorophyll. Chlorophyll, the pigment, color pigment, which absorbs sunlight. Chlorophyll. And because of this chlorophyll, these microorganisms, they can do what? Photosynthesis. They can take CO2 plus water plus what? Light. This light actually, this is the antenna right here that absorbs light, chlorophyll. Okay? And that is then converted into organic compound, glucose. Glucose.
It is present in cell membrane.
Cell membrane. These are the pouches, bags that are inside the cell membrane. This is the cell membrane, but it has little pouches right there. That is cell membrane. Yeah. Okay. So those are the three layers of envelope: capsule, glycocalyx, cell wall, and cell membrane. Chemical composition. What is this cell membrane made up of? Okay. Two things. Let's look at the diagram. Here. It's on page 89. Right here. Phospholipid bilayer, two layers of phospholipid, just like outer membrane of gram-negative bacteria. Okay, right here. Phospholipid bilayer, one layer, and another layer. And then proteins. Some proteins are structural, okay, just to hold the integrity of the cell membrane. Others are responsible for transportation. And these are called what type of proteins? Carrier proteins that are responsible for the transportation of materials in and out. They are very active in active transport. We will look at active transport later. And these proteins and phospholipids are not firm structures. They are loose. They are freely floating. Like if you take tennis balls right here and beach balls and throw them in swimming pool at nighttime, when you wake up in the morning, would you find each ball at its own spot? No. They will be moving. So this is called fluid mosaic model. It tells you that the phospholipid and proteins are fixed in the membrane. They're constantly moving. Why are they moving? Why are the proteins? Okay? Because the need of the cell, okay? On the need of the cell, what type of molecule is being transported? Small, large? Is it on the east pole or the west pole? Okay? Wherever the need of this carrier protein is, move towards. So they are not fixed. Okay? So now we move into the cell, we dive into the cell, done with appendages, three layers of the envelope. Now we are going to go inside the cell. The most important part inside the cell is cytoplasm, that all living cells have, cytoplasm. And most of the cytoplasm is what? Water. Water. No metabolic activity can take place without water. Okay? So, before I put this away, let me just show you the different parts that we're going to talk about. So, cytoplasm, inside the cytoplasm, what's the most important structure? Right here. Yeah, nuclear material. Yeah, prokaryotes have nuclear material or nucleoid right there, the purple part right here. Okay. In addition to, or can I say chromosome? Remember, this is only one chromosome. Looks like hodgepodge. Okay. This is not a very good diagram again. Okay. Why? Because this is not how This bacterium is 1 millimeter long, 1 millimeter. The length of this DNA is 1,000 millimeter long. The DNA of a typical bacterium is 1,000 times longer than the bacteria itself. How does it fit? Bacteria is 1 millimeter, DNA is 1,000 millimeter. Wraps around the histones, right? Sorry, Sandy. No. Bacteria don't have histone. Bacteria don't have histone. Yeah. Okay. It's called supercoiling. Okay. You take a rubber band. When you're bored, you start twisting. What happens to that rubber band? It's double-stranded. You're bored. You just keep on twisting. That's exactly how that long rubber band turns into a teeny tiny ball called supercoiling. That's how it fits inside the teeny tiny bacteria. Supercoiling. Okay. We'll talk about that in chapter 8 when we talk about DNA. Now, in addition to their own chromosome, many bacteria, they also have extra pieces of DNA. Do they have them here? No. Oh, they do, right here. This see circular piece of DNA right here? This is called right here. Plasmid. Extra chromosomal DNA. 3 types, right? If this is gram-negative, let's suppose if this is gram-negative bacterium, okay, it can have all 3. It can have F, okay, and F will allow this pilus formation right here, pilus, okay. Both gram-positive and negative, they can have the other 2 types of plasmids. The other 2 types of plasmids are M and R. It's in your notes. Let's go back to your notes. I just wanted to show you right here. Right here. Okay. Gelatinous substance mostly made up of water. Okay. And then it has amino acids and other sugars in it for metabolic activities. The most important intracellular structure, cytoplasmic structure / intracellular, you can also say. Everything that is you see right here, it is intracellular. Okay. Chromatin. Chromatin is combination of DNA, RNA, and proteins, but not histone proteins.
I'm sorry, did you say plasmids is all
Yes, yeah, intracellular. Yeah, everything that's listed here, chromatin. Okay. Actually, as a matter of fact, everything that's very good we are going to talk about from now onwards is intracellular. After this, everything that we will say is intracellular. Cytoplasm, ribosomes, plasmids, and everything. So our plasmid right here, which is a resistant plasmid, is found in both gram-positive and negative bacteria. Our plasmid, responsible for resistant to antibiotic, right? It is found in both gram-positive and negative bacteria. But this one right here, it is found only in gram-negative fertility or F plasmid. You may want to highlight it, underline it. F plasmid or fertility is found only in gram-negative bacteria. Okay. Both, don't write this, both females carry F plasmid, true or false? Both male and female gram-negative bacteria, they carry F plasmid. Only males have it, male. Because of the F plasmid, it is called male fertility because it has the F plasmid. It cannot conjugate with another female, unfortunately. Okay, or fortunately, pardon me. Okay. So, 2 negatives cannot conjugate, 2 males cannot conjugate, only male and female. So only males have, gram negative bacteria. Females don't, that's why that's female.
You said the female have F positive.
It becomes F positive after receiving it. After conjugation, the female will become male. Yeah.
Afterward.
Yeah. So my statement was both F positive and F negative, they have F plasmid. Negative, gram-negative. Yeah. So going back to my original statement, both F positive and F negative, they have F plasmid. True or false? False. Only F positive, they have fertility plasmid. F negative will become, after conjugation, it will become F positive. M plasmid, metabolic plasmid, both gram-positive and negative bacteria, they have metabolic plasmids. That allows them to gain extra characteristics. Okay. There was a very interesting experiment that was done by Army fellow by the name of Griffith. We'll talk about that in chapter 8. He proved that bacteria do pick up pieces of naked DNA from their surroundings. Okay? Naked DNA. They don't have to have conjugation because only gram-negative bacteria conjugate. So how come gram-positive bacteria have plasmids too, M plasmid and R plasmid? How do they get it? Other methods that we have not talked about yet. Okay? Ribosome, the second major intracellular structure, ribosomes. You need to know the size of the ribosome. Prokaryotes have 70S ribosomes, eukaryotes have 80S. Chemical composition. What is ribosome made up of? Two types of bricks. One brick is called rRNA, ribosomal RNA, and the other one is protein. So what do you need for the construction of ribosome? Ribosomal RNA and proteins. Okay. Inclusion bodies. This is one of the questions, I think, on your pretest. Okay. Inclusion bodies are intracellular storage areas. All living cells, not only prokaryotes, but all living cells, we store something in our cell. Okay. What is this store right here? Fat. Okay. But other cells, okay, they may store glycogen. Okay. They may store, some bacteria even store this. Look at this right here. Gas. Why do you think they need gas for? Very good, very good. You see this bacteria right here? Autotrophic bacteria. This is the bacteria you will most likely find gas vacuole in because it is autotrophic. It needs to float on the surface of the water. For what? To absorb light. Okay? So those bacteria that are autotrophic, you will find gas vacuoles inside them to raise them to the surface, close to the light. Glycogen is complex sugar, commonly used for what? ATP synthesis. This is a complex sugar. Sulfur. What do cells need sulfur for? Okay. How many amino acids are there? Naturally occurring amino acids. 20 amino acids. Out of those 20, there are 2 that have sulfur inside them. Out of 20, only 2 that have sulfur. If you don't have those amino acids, you cannot make the enzymes and the protein. So you need sulfur-containing amino acids. Those 2 sulfur-containing amino acids are called cysteine and cysteine. Very good. Cysteine and cysteine. These are the 2 stinky amino acids. That's why the eggs rot. When the eggs they rot, or you have rotten smell of sulfur. Okay. These are the ones that are being broken down. Bacteria use this to make hydrogen sulfide gas. What is the color of hydrogen sulfide gas? Colorless. May I have the atlas? We have atlas. I can. Yellow. Okay, let's see. Let me show you. Maybe then you can think. Yeah. This is hydrogen sulfide right here on page number right here. Okay, this is on page 58. 58. Right here. This tube has hydrogen sulfide. These 2 do not. So the color of hydrogen sulfide is brown. This is kind of blackish color, black. But don't write it. Don't write it. Okay. Now, If the color of hydrogen sulfide is gas is black, there should be a lot of embarrassed people walking around with black streak behind them. Have you ever seen that? No. It is colorless. It is. It was a tricky question. Okay. When you drive by a landfill, right? Landfill, you can smell it, but you don't see a black cloud. Darn, I have to run. Here comes the hydrogen sulfide. No. Okay. So hydrogen sulfide is clear. To make it visible, to make it more visible, so we can differentiate it from other gases, when it reacts with iron, ferrous ammonium sulfate, it turns black. So we have added iron to the medium. So any bacteria that makes hydrogen sulfide will turn black. Okay. Hydrogen sulfide, clear. Clear, clear. Yeah. Thank you. All right. All right. Last topic, I think this is last. What is this? Oh no, oh yeah, one more. And the last topic of this chapter 4 is the last intracellular structure that we will talk about. There are others that I'm not going to talk about. It is endospore. Endospore. Okay. What is endospore? Okay. Metabolically inactive structure. The key, another key question, it is inactive. Here's the key question right here. Non-reproductive. The purpose of endospore is not reproduction. When one endospore germinates, okay, you only get one vegetative cell. Yeah. Okay, let's see. Here we go. Picture on page 96. All right. Right here. Now, when the conditions, environmental conditions are unfavorable, temperature may be a little bit too high, no food, no moisture. 5 bacteria, the name is coming up, no names of those 5 bacteria, they can do this. Okay. Now, tell me please, what will happen to this cell when the conditions are favorable, young culture, lots of food? It's going to undergo binary fission. One cell will split into 2 identical cells. Okay, you see, that's actually how the binary fission starts. But when the conditions are bad, here's mommy cell. It makes a copy of the DNA right here. And remember the names of the 2 chemicals? She starts to deposit dipeclinic acid and calcium around this. So this copy of the DNA remains metabolically inactive. Now let me repeat this one more time. Question on the test. Make sure you understand this. Is this a bacterium right here? Metabolically active. Mommy is alive, right? Mommy will keep functioning. The copy, she's pregnant, all right? Okay. She has not given birth yet. So it has DNA and it will keep on functioning. It will keep on functioning as long as there is food. So my question to you is it possible for a vegetative cell right here to have an endospore? Yes, it's right here. This is a vegetative cell, metabolically active cell. Can it have an endospore? Sure, right here. Very good. Remember when you did endospore stain and I showed you endospores inside the vegetative cell, red cells with green spot inside them. Okay, so that's my question. The question is it possible for a vegetative cell to have an endospore and still be metabolically active? Yes, right here. Okay. Once the environmental conditions become extremely unfavorable, mommy dies right here. Mommy is gone. The endospore is now released. So this process right here, these are in your handout, I think the 2 terminologies. One is called sporulation or sporogenesis, a process in which metabolically active vegetative cell transforms into a metabolically inactive endospore is called sporulation. One more time. A process in which a metabolically active vegetative cell transforms into metabolically inactive endospore. Under what? Under unfavorable growth conditions. One more time? Okay. Sure. A process in which a metabolically active vegetative cell / bacterium transforms or turns into a metabolically inactive endospore under unfavorable growth conditions. Okay. Now this can remain dormant for how long? Centuries. That's why they don't use E. coli and salmonella and biological warheads. They use spores, spore-producing bacteria. So even if the pilot misses the target and the bomb is dropped in a deserted area, somewhere there's no one there, after 10,000 years, that area is populated, the spore will still kill. Okay? Yep. Mm-hmm. In the mummy, yeah, that was non-pathogenic. It's a type of bacillus. Bacillus, non-pathogenic. Yeah, non-pathogenic. Yeah. Mm-hmm. So the opposite, when you take the endospore, put it in favorable growth condition. Okay, now this was the question, I think. If you have one endospore and you put this in a fresh broth, fresh liquid, how many vegetative cells will come out from this endospore? One. That's why it is not a reproductive structure. Okay, it's not. The purpose is to save copy of the DNA, non-reproductive. So one endospore, one vegetative cell. So the opposite of sporulation is called germination. Germination. And you can just flip the terminology. A process in which metabolically inactive endospore transforms into metabolically active vegetative cell. Under what condition? Favorable growth conditions. Okay. Here's a seed. You put it in the ground, put the moisture, put the water on. It's going to turn into a tree. That's what's going to happen. One endospore, one bacteria. Now, the memorization part. The 5 bacteria that make endospore. Okay. So both gram-positive and negative, they can make endospore, and here are the names right here. 3 gram-positive, the first 3, any bacterium whose first name is Bacillus, Bacillus cereus, Bacillus subtilis, Bacillus anthracis. So all bacteria, okay, if this name starts with Bacillus, it's going to make endospore. Clostridium, Clostridium botulinum, Clostridium perfringens, that causes gangrene, tetanus and all that, they all produce endospore. Okay. This one, I believe, is non-pathogenic. I have not heard of any disease this one causes, but this is also gram-positive. Sporosarsina. Sporosarsina. So these are 3 gram-positive, 2 gram-negative. D-sulfotomaculatum. Yeah, say it 10 times faster, right? Yeah. Sulfotomaculatum, gram-negative, and Oscillospira. These are the only 5 bacteria that can make endospore, none of the others. Go ahead. Non-pathogenic. Those 2 are non-gram. Yeah. The first 2 are not all, but yes, there are Bacillus species there are some species of Clostridia that are, yes. Yeah. Yeah. By the way, all these spores, where do you find these spores? Everywhere. Ubiquitous. They are everywhere. Everywhere. Wherever you find dust, you'll find spores. Okay. But we will talk about this later, or maybe you should address this. Like Clostridium botulinum, Clostridium perforans gene that causes gas gangrene. That bacterium is obligate anaerobe. Anaerobe. Okay. Okay. First of all, not only you have to remove oxygen, you have to give that bacteria extra 5 to 10% carbon dioxide.
So people that are diabetic,
Diabetic, yeah. This is what happens to them. Gangrene. How? Okay. Their work. Loss of circulation. That's the key word. Very good. Loss of circulation. Yeah. This is what happens. You see, because of sugar concentration, they lose their sensation in their extremities, in their fingertips and toes. So even if they cut themselves, they're bleeding. They do not realize that they have injured themselves. And these spores are in the soil. So spores, if they end up into the dead tissue, dead tissues, they give off CO2. Okay? That's how the gangrene starts. Yep.
is that because of being hypersonic, because you have so much sugar, it's creaming, the cells are being creamed?
No, that's a different thing. If your blood sugar is too high, most bacteria will not be able to grow. Most fungi, they are osmophile. Any bacterium, any microorganism that can tolerate high salt concentration or high sugar, it is called osmophile. So diabetic people are most likely to get fungal infection, most likely. Why? Because fungus can tolerate high sugar and salt concentration. Most bacteria cannot. But gangrene is a different thing. Gangrene bacteria, my question I was going to ask you, which part of our body, normal, in a normal human being, which part of the body can facilitate or that can provide 5 to 10% CO2? So it can grow. None. If you have 5 to 10% CO2, carbon dioxide in your body, carbon dioxide, you're not alive. Should not be. That part should not be alive. So gangrene, that's why gangrene is what? Dead. Okay? Black tissue, dead. Now, in a normal, healthy human being, there's no organ, no part of your body that has 10% CO2. You see, whatever the big boss does, there's a reason. These spores are everywhere, and you're inhaling them right now. So why don't you get sick all the time? Because we are aerobic creatures. Aerobic. And these are, at least many of these right here, Clostridia are anaerobe. Okay? Clostridia, many of them are anaerobes. Okay?
On the study aid, Clostridium, you have for gram-positive and gram-negative?
No, it should be gram-positive, not negative. Positive, yeah, positive. It should be gram-positive. If it is, please correct it. It is gram-positive only. Yeah. This is the end of chapter number 4. We will start chapter 5. How much time do I have? 20, well, that's a whole lot of time. So we will, yeah. So we could have taken the test on Tuesday, guys. Fine, I understand. No problem. The exam is only on chapter 1, 3, and 4, not on 5. Okay. Okay. I did not bring my PowerPoint presentation, so I have to go to the old style notes on my web page. What? Don't tell me. Okay. All right. Chapter 5, microbial metabolism. You know, the other class is taking the test on chapter 1, 3, 4, and 5, and they're taking it today. Yes, they are. Professor Apter's class is taking the test today on chapter 1, 3, 4, and 5. Yep. Okay, anyways, okay. Metabolism. Okay. In this chapter, we are going to talk about basically how do bacteria make ATP? How do bacteria eat? How do they transport nutrients? What is required for each process? What is made by each process? What is released by each process? Metabolism. Chemistry, intimidating, but I will try to make it as simple as I could. Okay. How do you define metabolism? This is one definition, sum of all chemical reactions. Okay, there are two types of chemical reactions, anabolic and catabolic, okay, in any living cell. How else would you define metabolism?
Breakdown and synthesis of
Yeah, breakdown of complex molecule into simpler forms and vice versa. That's fine. Okay. What else? Balancing act. That's all it is. Your body is trying to balance. The Chinese proverb, yin yang, right? Yin yang. Good energy, bad energy. That's exactly what metabolism is. Your body is trying to balance itself. It's right here. Energy balancing act. Okay. Yin yang. Okay. On exam 2, you do have a question on this terminology, anabolic or synthetic processes and catabolic. Okay. Anabolic process is a process in which you take smaller molecules and you build large molecules. You have glucose, you add another glucose. What do you have? Maltose. Now, whenever you create a new bond, you lose one water molecule. Water. And that's called dehydration synthesis. Synthesis means building up. D means what? D, removal, without. Dehydration. Hydration means water. Dehydration synthesis, a process in which water is released to build a larger molecule. Dehydration synthesis. Okay? Now you drink coffee or you drink tea, your blood sugar level is low. Instead of synthesis, which is right here, is going to break down. And that is called catabolic reaction or hydrolysis. Hydrolysis means using water. Now this time you have to return the water back. So hydrolysis, hydro from water, lysis means breakdown, hydrolysis. Or catabolic reaction. Okay. Synthesis is anabolic. Okay. Catabolic reaction, they use water. Anabolic reaction, they release water. Okay. Question, just to make sure you understand it. D means removal. D, yeah, release water. It does. It does release water. D means removal. Psst, removal. Okay. So you have 5 glucose, 5 monosaccharides in a polysaccharide. Okay. You have 5 monosaccharides. You have synthesized a polysaccharide. How many water molecules were lost during the synthesis of this polysaccharide? How many? 4. Fausia says 4. So we have one answer. 4. 1. 1. Any other answer? Okay. 20?
3.
Oh, 3. 3. Okay. Okay. Now, once I tell you how to do it, it is so simple. Okay. Let's draw 5 molecules of glucose. Glucose, 2, 3, 4, 5. And for each bond, you release 1. 1, 2, 3, 4. So here's a little trick. Whatever number I give you, minus 1. Okay? Whatever number I give you. 5,000 glucose molecules in a polysaccharide. How many water molecules? 4,900. There you go. Minus 1. But on the test, because I am a little tricky, okay, I may give you water molecules. This is still the same principle. I tell you 10 water molecules. 10 water molecules were released during the synthesis of a protein. How many amino acids are in that protein? 11. If I give you water molecules, you add 1. Very good. If I give you the building blocks, you subtract 1. If I give you water molecules, you add 1. All right? Make sense? So, 49 more questions for exam 2. Okay? Let's see. So that's DNA. No, wrong. 4. Okay. That's wrong. Fine. All right. So the 2 examples. One example, okay, you have, this is all messed up. Let me see if I can fix it. No. Okay, fine. You have 2 amino acids. You should have an arrow here. Draw an arrow. You have a dipeptide, right? And you lose 1 water molecule. That is dehydration synthesis. Or you can have 2 glucose, the one that I just did. Glucose, amino acid, doesn't matter. 2 smaller molecules to make a larger molecule. Bacteria, when they are under good, favorable condition, what do? They synthesize capsule. Synthesis of capsule. And what is capsule? Protein or sugar. So they take these building blocks and they build up their capsule. So building of capsule is process of synthesis, anabolic reaction. Catabolic, opposite. Or it is also called hydrolysis. Using of water to break bonds. Hydrolysis. Most catabolic reaction, okay, they release, not all. There are a few exceptions that I'll give you the names, okay. Same thing. This time you have dipeptide. You are going to add water. This is all, sorry, messed up. Breakdown of capsule under unfavorable condition. When the conditions are unfavorable, what do bacteria do? They eat up their own capsule. Okay. Now for each reaction to take place, anabolic or catabolic, okay. For example, I give you Metamucil, okay, in 2 tubes. You have 2 test tubes, and each test tube you have Metamucil. Okay. In tube A, I add a drop of cellulase, the enzyme. In the other tube, no enzyme at all. No enzyme. How long would it take for cellulase to break down these carbon into individual sugars? How long? Within seconds. Within seconds. Enzyme will work on the substrate, and these bonds will be gone. Okay, right here. All right. No enzyme tube. Do you think these bonds will be broken down without enzyme? Thousands of years. You come back, you ask your grandson, please check that test tube that I left, okay? Yeah, maybe some of the bonds will be gone without enzyme. That's how important enzymes are, okay? So that's how you can digest your food so fast. Okay, some people that cannot digest lactose, ask them, they have to take the help of the bacteria or enzymes to digest them, to help them digest substrate. So enzyme, what do? They are all proteins, first of all. All enzymes are protein. They change the rate of the reaction without being changed themselves. Enzymes, you can call them organic catalyst or biological catalyst, same thing. Okay. Protein produced by living cell that change the rate of the reaction. Important thing is that they are not used during the reaction. After taking our reaction, enzyme can be reused over and over and over again. Okay. What's the difference between an enzyme and a hammer? Crystal? Okay. Anybody? Enzyme and a hammer. I know it's a weird question, but
It's targeted. Like we don't have,
targeted, direct,
we don't have to use any
energy. Okay. Good answers. Okay. Yeah. One hammer. I give you one hammer and ask you to please go ahead break this table. You can do that, right? And ask you to give you the same hammer. Ask you to build something with that same hammer. Can you do that? Sure. Multitasking. Hammer can do multitasking. Enzymes cannot. That's the answer I'm looking for. Okay. One enzyme, one function. Cellulase will work on what? Cellulose. It will not, although starch is made up of the same thing, it will only work on cellulose, not on starch. You need a different enzyme for starch, amylase. Specificity, that's the answer. Enzymes are specific. One enzyme, one substrate. If you want to break down your table sugar, sucrose, can you use cellulase? No, you need sucrase. Sucrase. Specificity. Okay. Substrate. What is the substrate? Anything on which an enzyme works. Any substance on which the enzyme works is called the substrate. Cellulose, substrate. Gelatin, substrate. Potato chip, substrate. Yep. What enzyme do you need to break down potato chip? Other than your teeth, those are not enzyme, but they're mechanical. After it's done here, what enzyme? Amylase. Amylase is starch. Starch. So amylase. It starts right from your mouth. Starch. Okay. So any substance on which the enzyme works. Now this enzyme, here's the enzyme, here is the substrate. Can I insert this substrate anywhere in this enzyme? Okay, this is enzyme right here. Here is the substrate. Can I just put this anywhere, or there's a special area here? Special area, right? This special area, this specific receptors which is located on enzyme is called active site. They have another site in the back. We'll talk about that later. Many enzymes, they have another area. Okay, but this is where the site where enzyme reacts with the substrate is called the active site. Catalytic site, active site, same thing. Specific portion of enzyme that reacts with substrate. Oh. Pardon me? Oh, 5 minutes, thank you. Okay. Enzyme. Substrate. Sometimes you may have more than one substrate that looks alike. I think I may have mentioned to this class or different class. This is called molecular mimicry. If you want to kill this bacteria, the enzyme is inside the bacteria. You want to kill this bacteria. This is bacteria's normal substrate. All bacteria, they use this chemical. Okay? We cannot do that. Okay? That's why when you're pregnant, one of the very first things that the doctor may give you is folic acid to facilitate the growth of the fetus, the baby. So bacteria, they make their own folic acid by using this chemical right here. So this chemical goes in, and the bacteria make folic acid. But you're sick. This bacteria is making you sick. And the doctor wants to chop off this bacteria. There are several options. Okay, you can take penicillin, different mode of action. But one of the options doctor has is to give you a medication that looks like PABA. And that is that drug. You have heard of sulfur drugs, right? Many people are allergic to sulfur drugs. That's the same thing. But sulfur drugs, they look like PABA. So you take a medication, sulfur. Molecular, they're similar. If sulfur goes right here in the active site, bacteria is dead. That's how AZT works too for HIV, AZT, molecular mimicry. Okay? I'm going to stop here. We'll pick up right from here next time. Good day.